Yes, please.
Okay, so first of all, I would like to thank the organizers of this meeting for the invitation to present this talk today. Also, I would like to welcome all of you online attending my talk, and also congratulate everyone to take part on this meeting. So today I'm going to talk about the impact of the nanomaterials morphology in cell viability assay. So this is the topic of my talking today. So here is the outline of my talk. Briefly, I will cover some details about the literature search, search on in vitro and in vivo bioassays. Then I will show you the typical data you can find these days in the literature. And then one of the two important topics of my talk, I will talk about the Hue model. And next, how can you apply the Hue model to describe and understand most of the in vitro assays? And then the second important topic, the second most important topic of my talk is how can we use the Hue model to include the nanomaterials morphology and extract from these the impact of the nanomaterials morphology on in vitro assays. And then the next topic is how to use this proposal of including the nanomaterials morphology to explain the in vitro assays. And finally, how we can simulate interesting data based on this Hue-inspired model. So, about the literature search, if you go, for instance, Web of Science, and check the number of in vitro and in vivo assays, publication involving in vitro and in vivo assays, you will find roughly about the same number in this platform. So it's about 3.5 million papers published. But if you look closely to this publication, you will find about only 0.2% of those papers, including any kind of mathematical model to explain the data. Of course, this is a very, it's a big surprise. The good point about this so small number of papers with including a quantitative analysis. The good point about this is that in the last 2 decades, the number of papers including any mathematical model is increasing over linearly, which is a very good hope for the future. So here is the let's say big challenge: how to move from a typical qualitative analysis of in vitro. Let's focus on in vitro now. So how to move from a simple qualitative analysis to a more robust quantitative analysis. You need to go through 2 main steps. The first one is using the curve fitting to be able to fit the data. And, well, this is a very old proposal, actually, from the second part of 18th century. It's called the least square fitting. Remember, the guy who introduced this was Gauss, Carl Friedrich Gauss. So we have this tool for more than 200 years. The second point is describing in vitro assay using a mathematical model. The very first successful mathematical model for explaining in vitro assays, it was proposed in 1910. It's already more than 100 years. So the combination of these two tools allow us to move from a simple qualitative analysis to a quantitative analysis. Nevertheless, the number of papers including this quantitative analysis is, let's say, well below 1%. But it is increasing, fortunately. Okay, so let me show a typical in vitro data presentation. I just took one paper from March this year, March 2024. But you can scan, you can search, and you can check by yourself that most of the papers published on evaluating cell viability, they are presented in this typical, I will call, semi-quantitative analysis. So here we have one experiment that they were testing a chemotherapeutic agent. So the chemotherapeutic agent is the echinacin. And they used cell lines to test the cell viability or the cytotoxicity of these two cell lines when they are challenged by this chemotherapeutic agent. Okay, so you can see that the vertical axis is the cell viability in percentage. On the horizontal is the concentration of the chemotherapeutic agent. The difference from the left and the right hand panels is the cell line they used. And they incubate the cells at increasing concentration of the chemotherapeutic agent. And the test was performed leaving the incubation for 24 hours, 48 hours, and 72 hours. So what is the big point that I want to emphasize here is that symbols here in both plots are experimental data. And the solid lines are just straight lines linking the experimental points. So, of course, you can extract some information from both set of data. But the information you can extract is, let's say, sort of semi-quantitative data. You can say, okay, the data extracted after 72 hours present lower cell viability in both cases. But nothing beyond that. Not really a robust information you can extract. You can, for instance, using this data, you can say something about the lethal dose 50, the LD50, or the benchmark dose. But it is only a, let's say, sort of very good qualitative analysis, or I can say semi-quantitative analysis. So we want to using a mathematical model and using the curve fitting, we want to manipulate data and extract from this manipulation precise information. Then, to perform this, the Hill model was proposed in 1910. So it was proposed by Archibald Vivian Hill. This paper was the second paper Hill published in his life. And it is interesting. The paper was published in 1910. And only 12 years later, Archibald Vivian Hill received the Nobel Prize in Medicine and Physiology. So this paper has been used for more than 100 years, very successfully in many, many cases. Then, let me show you how simple the model is. Here is very briefly how the Hill model was deduced. So you have a receptor, let's say, on the cell membrane, and you have a molecule which is going to bind to the cell membrane receptor. And you can represent this binding by a sort of equilibrium process. On the left-hand side, you have the receptor. You can imagine it on the cell membrane. And then there is a number of molecules that can bind to the cell membrane. N is the number of molecules that can bind. And then this binding produces the complex. So the molecule binding to the cell membrane receptor. And we can represent this as a sort of equilibrium. And then we have the forward constant of equilibrium and the backward constant of equilibrium. So the standard way to describe the forward constant or backward constant is in terms of the concentration of the species on the left-hand side and the right-hand side of this process. So here I represent the backward constant. The backward constant is the ratio between the product of the species on the left-hand side, and the exponent here is the coefficient of the process over the concentration of the complex. So this is the backward constant. Another point we should emphasize in describing this process is the fraction of the bound structure. So this is the complex or the bound structure. What is the fraction of this in the system? The fraction of this in the system we call Fn. It is the concentration, here's the molar concentration actually, it is the concentration of the complex over the total concentration of receptors. So receptors which are not bounded and receptors which are bounded by the molecules. So this is the fraction. Of course, I can combine these two equations. I can extract the concentration of the complex in the first equation and replace in the second equation. And then I can describe the fraction in terms of the concentration of the ligand molecule. So from this, I can describe the cell viability. So the cell viability, we call V, depends on the dose. D is the dose, and D actually is the concentration of the molecule. And then the cell viability is how much the fraction deviates from the starting point. So here's the cell viability. V naught actually is the cell viability when the dose is 0. So it's the pure cell line. So this is the standard Hill equation. You see that the concentration of the molecule is replaced here by the dose. And we have the exponent D. The exponent N, the exponent N is called Hugh coefficient. And look at the backward constant where it was replaced here by the C constant. So this equation is a very nonlinear equation, and it's very useful to explain most of the cell viability data. Okay, we have two very important situations here. One, both are related to the number of the Hugh coefficient. If the Hugh coefficient is over than 1, we call this positive cooperativity. If that is lower than 1, we call negative cooperativity. What that means in practice? In practice, it means if N is over than 1, first molecule that binds to the cell membrane facilitates the binding of the next one. On the contrary, if N is lower than 1, the first molecule that binds to the cell membrane just blocks the binding of the next one. So this is a very important border. Okay. Let me show you one application of the standard Hugh equation. Here, we made this analysis in a recent paper. So, in this case, we used the cell line, a very famous cell line, the HeLa cell line, and we performed the cell viability using the NTT assay. And we challenge the cell line with graphene quantum dots, but graphene quantum dots doped with fluorine. We performed this test aiming at the application of fluorine doped graphene quantum dots in photodynamic therapy. So, in other words, fluorine containing graphene quantum dots is here tested as a photosensitizer. And we performed then the cell viability assay in two different conditions. In the dark, or illuminating the cell line with red light. So here in the plot on the left-hand side, you can see the vertical bars are experimental data. The black ones are the data collected in the dark, and the red one are the data collected under illumination with red light. So this particular sample we analyzed, you can see the TEM micrograph of this sample and the particle size distribution. It's around 2 nanometers in diameter, actually. On the corner here, you can see the high resolution TEM, just to check the interplanar distance and check that the phase we were working was exactly graphene quantum dots. So, of course, different synthesis, we come out with a mean particle size slightly different, but not far from about 2 nanometers in diameter. Okay, so the fitting here, using this the Hugh standard model, are the solid lines. You can see the black solid lines, a very nice fitting, and the red solid lines, also a very nice fitting. From this fitting, we can extract with more precision the DL50 and the benchmark dose, for instance, which are very important parameters. Okay, so the question at this point is, if I want to include the nanomaterials morphology, for instance, the mean size and the size dispersity of one particular nanomaterial, keeping the phase and keeping the shape, such as, for instance, the graphene quantum dots. So I will keep the phase, which is graphene, and I will keep the shape, which is small islands, flat, small, round islands. Okay, so what happens to the cell viability if I change the mean size and the size dispersity? So the first important question is the standard Hill model a promising starting point to include the nanomaterials morphology? And the second point, if the Hill model is a good starting point, how to start from it? Where should I work on the standard Hill model to step up in a way to include the nanomaterials morphology? So here is a standard mathematical representation of the Hill model. And we are going to work on the Hill coefficient to include the nanomaterials morphology. How to do this? Here's a scheme that I want to show and convince you that this is a fair proposal. So here is, let's say, a typical picture of a nanomaterial. And on the left-hand side, the green, I represent the cell and the cell membrane around it. So, of course, the nanomaterial can bind the cell membrane different points, 1, 2, 3 R points or N points. Let's say nanomaterials has N points for binding, but it can bind to the cell membrane only using R, where R would be less than N. On the other hand, you can also analyze the system looking at the binding of the nanomaterial to biomolecules. All systems should work. So now, how can represent the binding between this nanomaterial and the cell membrane? I represent, again, using the same scheme that Hill used in his standard model. So there is a R number of points on the cell membrane receptors binding to endpoints available on the nanomaterial. Nanomaterial is the ligand. And then this is the complex. And again, you have the backward constant and the forward constant. You can calculate the backward constant, same way as before, and then you can calculate the fraction of the binding structures. And you come out with the same expression, except now we have the Hill coefficient. And now the Hill coefficient is not constant anymore. So I did expansion, first order approximation of the Hill coefficient. Here is a standard first order approximation. So it is a straight line actually. So then you have the basic or starting point and the difference between the size of the material and, let's say, the mean size times the coefficient. So this is a straight line. Okay, now I need to include, because this describes the size of the nanomaterial, I have to include this expression on the fraction. And then, of course, I need to use some statistics to describe the nanomaterials distribution. It could be, for instance, this one here, which is the log normal distribution function. And then I need to calculate the mean size or the mean fraction. How do I calculate the mean fraction? It's the convolution of the fraction and the particle size distribution. Okay, so now I can describe the mean cell viability. Same way is the value of the cell viability at zero concentration of the nanomaterial minus the fraction of the nanomaterials. So this is a standard way. If I do this, I can use this expression to fit the data. Let me show you interesting results about this. Okay, so here is the data I showed before, which is the fitting of the cell viability using a standard Hill model. I took the experimental points here under dark with just the black spots and this plot in the center of my slide, and the experimental data of the cell viability under illumination, which are the blue spots here. And then I did the fitting using this expression, which includes the nanomorphology of the material. You have the mean size of the nanomaterial, and you have the size dispersion. So the mean size is the mean of X, S, and the size dispersity is sigma here. Okay, you can see the black solid line is the best fitting, and blue solid line is the best fitting. But you may say, okay, fitting on using the extended Hill model and the Hill model, it looks about the same. Well, the point is what you can extract from this fitting. So we can extract the mean size and the size dispersity. And we can compare these with the data extracted from the TEM. So TEM is evaluation of the physical size. And the TEM of this give us the mean size of the graphene quantum dot is about 1.98 nanometer, and the size dispersity is 0.19. So, and then what we can extract from this fitting is not the physical size, but is the size that was probed by the cell line. I call it the biological size. And I have a biological size coming from the dark experiment, and I have the biological size coming from the light experiment. And it's very interesting, although the mean size are roughly about the same, but they show very interesting trends. The interesting trend is under dark, the mean size is a little bit lower than the physical size. So the cell sees the nanomaterial a little bit lower in size than the physical size of the nanomaterial. However, if you illuminate, you increase very much the size toxicity, and it reflects on the mean size that the cell can seize. So it's a little bit lower, 1.77. Although the size dispersity are within the error bar, actually. And then this gives us a very important information. The new information is what I call the biological size. And the biological size is a way to understand the cell toxicity when it is challenged by a nanomaterial. Okay, now I can simulate, using this Hill-inspired model, I can simulate some trends, important trends. For instance, here in this simulation, I fixed the mean size at 2 nanometer, which is about the size, the typical size of this nanomaterial. And then I increase the size dispersity from 0.05 to 0.50 under light and under dark. And what I found in both cases, if I decrease the size dispersity, which means I am taking the sample and the sample is going to more and more monodisperse. 0.05 is really monodisperse. So you can see if I reduce the dispersity, the cytotoxicity increases. So lowering the size dispersity increases the cell toxicity. I can do the same analysis by simulating what is the impact of the mean size. So here I fixed the size dispersity in 0.3. And then I want to explore how the system develops if I increase the mean size from 2.1, which is typical of the sample we analyzed, up to 5 nanometers. And what I found that as I increase the mean size in this range, this is a very important point, at very small range, below 5 nanometers. So if I increase the mean size from 2.1 to 5, what I found is that I increase the cytotoxicity. So if you go to the literature and do a search, you will find a lot of papers published and only containing experimental data showing that the cell viability depends on the sample's morphology. If you change the mean size or change the size dispersity, the cell response also changes. So, and this is actually the first proposal to take into account the nanomaterials morphology in the evaluation of the cell viability. So this was a publication about 1.5 ago. So I'd like to thank you, all of you, attending my talk. And again, thanks to the organizers. Thanks my two universities, the Catholic University of Brasilia and the University of Brasilia. They are different institutions. The Catholic is private, and the University of Brasilia is a public university. Here is my email. If you want to contact me, please feel free. And now, if you will, I can take any question from you. Thank you so much.